I don't think you can see it here, but this was a PCR assay for IAP DNA. So one of the things we'd like to do with XMRV is develop an animal model that recapitulates infection. This was a presentation given by Francois Villiger from Emory University, where they have infected rhesus macaques. The issue is, will it replicate in a model close to man? And if it does, what are the viral kinetics, dissemination, and the antiviral immune response? And so in their study, 5 macaques were actually infected. Blood, semen, and urine was collected at different times. There was reinfection here. Interesting, there was an immune response. It could bring a virus under control several weeks later. What came out of this study is that rhesus macaques are susceptible to XMRV infection. It results in a chronic, persistent infection, even though the viremia appears transient, with low viral loads within the first 10 to 21 days post-infection, followed by undetectable viral loads. Infected bone marrow-derived cells appear to be of lymphocytic lineage, T and NK cells. The lineage of the cells positive for XMRV infection replication in lymphoid organs are CD4 T cells, and other organs, and those of the reproductive tract, liver, pancreas, etc., appear to be either epithelial or interstitial cells. In the lung, most of the infected cells in the lung appear to be alveolar macrophages. The primary foci of infection in epithelial cells in organs such as prostate, seminal, vesicle, and epididymis during early and chronic infection. And chronic infection appears completely disseminated in select animals with extensive numbers of fluorescence and pseudohybridization and immunohistochemistry positive cells, even in monkeys exhibiting negative plasma viral loads, suggesting a low level of viral release. So the rhesus macaque is a model that can quite accurately recapitulate XMRV infection. It's also a very expensive model. And obviously the alternative models, such as mouse models, that could recapitulate XMRV infection would be desirable. There was one report from Dr. Ikeda from the Mayo Clinic on Mus pahari. I had to look this up. Mus pahari is the shrew mouse. And Dr. Ikeda reported that Mus pahari fibroblast cells support XMRV replication. Proviral DNA is found in the blood, spleen, and brain, suggesting that XMRV is both lympho and neurotropic in Mus pahari. A subset of infected mice showed panleukemia or pancytopenia. Infection of Mus pahari with XMRV rapidly elicits a humoral immune response. Again, we see this hallmark of G to A hypermutations, and this is a result of the mouse ApoBec3 protein. Dr. Ikeda is proposing that Mus pahari can be used to study the pathogenicity of chronic XMRV infection. Dr. Ikeda also presented a slide showing some of the problems of using Mus pahari. This is a very small mice. This slide does it a real favor here. It's a very, very small mice. Apparently, they're quite aggressive. When you try to get hold of them, they shed their tail cells, and they also pretend to be dead. So that aside, Mus pahari is actually a reasonable model for recapitulating XMRV infection. We also thought, when I was on the organizing committee for this meeting, we also thought it'd be nice to talk about vaccine development for XMRV. And I asked Ellen Sparger from the University of California, Davis, to give some thoughts on an XMRV vaccine. And the reason I asked Liz was that for several years she's been working with feline leukemia virus, developing vaccines for feline leukemia virus. And FELV is also a gamma retrovirus that was first isolated in 1964. There are endogenous and exogenous FELVs. FELV infects domestic cats worldwide. It's a common clinical entity and cause of disease in domestic pet cats. This is the reason we brought Liz over to talk, that the incidence has been significantly reduced due to screening and vaccination. And one point that Liz made was the similarities between FELV-associated and XMRV-associated disease, including immune suppression, a disease of the central nervous system, polyarthritis and joint pain. They both induce cancer. And so the lessons that Liz gave us from FELV, I think we would be well born should we get this far with XMRV in terms of vaccine development. These are the results from the vaccine studies that have been done with FELV, several methods of vaccination, ranging from whole inactivated virus to experimental DNA vaccines. We see efficacies ranging from 44 to 100%, almost complete protection with the new experimental DNA vaccines. But again, Liz made this point that the pathogenesis and immune correlates for XMRV must be characterized to inform any vaccine design and the use of novel technologies. So this is going back again to the Lowe and Alter paper. This PNAS study showed that it supports the finding of MLV-related gene sequences in the blood of patients with CFS. The viral sequences could also be detected in a small number of volunteer healthy blood donors. They differ from the reported findings on the near-genetic identity of all XMRV, inasmuch as patients with CFS, prostate cancer, and blood donors, analysis of the viral gene sequences revealed a more genetically diverse group. However, I want to bring you back to one of my earlier slides, and this was a very important point made here when we were asked, where do we go from here? One point was made that we have no sequence of a full-length viral genome in these studies, and we have no evidence for a replicating virus. And so until we have both the full-length sequence and an infectious virus, I just wanted to reiterate this point that the study of Urisman and Lombardi, where they detected infectious virus, and this study where they're detecting viral fragments, really have to be considered as two distinct phenomena. And again, we went back to this issue. Dr. Lowe from the Alter group reiterated this issue, the main sources of contamination, the PCR amplicons. This was, again, I don't have any slides for this, and I can maybe take a couple of minutes. This was another theme that was brought into the meeting, the source of contamination. The amplicons, the primers that we use for PCR identification, these can be a source of contamination. And at the HIV drug resistance program in Frederick, we went through a very testing time when the amplicons or the primers we ordered for a company came contaminated. And now our level of false positives showed up. The amplicons themselves can be contaminated. Robin Weiss recently reported in Retrovirology, I believe, an enzyme that was contaminated with mouse DNA. In the course of my year with XMRV, I got an email from Japan from a researcher who again said that the batch of enzyme that she was working with, because it contained antibodies that were made in mice, it contained mouse DNA and was testing positive. We talked about the use of biologicals, whether when heparin is included, is heparin a potential source of mouse DNA? And this can go on and on into the basic chemicals. We don't know how the basic chemicals are stored. And we don't know if these have been contaminated with mouse DNA, but we have to take this very seriously as people are finding polytropic sequences, potential mouse DNA, potential intracisternal A particle DNA.